Well, good evening, everyone, and welcome to my session. Well, actually, you may understand today what I'm going to discuss by seeing the diagram of the whiteboard. Well, actually, excretion. Excretion is the process by which waste products of metabolism are removed from the body. And the kidneys are the main excretory organs of the human excretory system. Well, actually, in my last class, I have discussed about the urinary system, the structure of kidney, and the diagram of nephron. And today I'll be talking about something details and something more about nephron. What happens actually inside the nephron or in the nephron? Well, well, so each human kidney contains almost 1 million nephrons. Understand? And the blood plasma leaves the blood in the Bowman's capsule and enters in the nephron. And then actually what happens in the nephron? Let's talk about that. So before explaining the filtration in the nephron, I'm just going to show some of the different parts of the nephron, though you know, but today just I'm uh, telling. See, I do not know you may see clearly or not actually. So this is glomerulus. You know very well, I think. And blood in and then blood out. Okay. Then Bowman's capsule. Then you see first coiled tubule. Then second coiled tubule. And then in the middle, the first coil and second coil tubule are separated by a long loop that is called loop of Henle. Loop of Henle. And then there are other or several nephrons joined here to form the collecting duct. And then this is duct opening at the tip of pyramid. Understand? So you are also seeing there I have just marked here this nephron. So this is medulla and this is cortex. Cortex means I don't know you have remembered or not. Oh, uh, Sarah Jerin Tasnim. Hello, Sarah. How are you? I'm really very happy by getting you. Okay, keep on watching. So, what was I telling? This is cortex. Understand? So, cortex, if you know the structure of the kidney, so you will be well known about that. See, the cortex means if we cut a kidney lengthwise, on that time we will be able to see this structure actually. This is a darker outer region of the kidney, and this part is cortex. So actually, this is cortex. Okay, and then it just contains some microscopic, though the cortex contains many tiny blood vessels branch from the renal artery. Okay, but it also contains some microscopic tubules or kidney tubules or a filtering unit. So um, some filtering unit. These are called actually nephron, and these nephron just run down through the middle layer of the kidney. This is called medulla. So this part is medulla, and the darker outer region is cortex. Understand? Okay. Then, well, so let's talk about the changes to the filtrate in the nephron. Okay, so now let's talk about that. There are two coiled region in the nephron. Two coiled region of the kidney tubules in the nephron are separated by U-shaped loop that is called loop of Henle. Understand? And then after second coil, this is first coil tubule and this is second coil tubule. Then after second coil tubule, there are several nephron joins together to form the collecting duct, where finally the urine passes out into the pelvis. Okay, now I'm just going to the main point that is the motto of my today's discussion. I mean the ultrafiltration of the nephron, because this structure already I have shown in my last class. Okay, so uh, I'm not understanding what class are you in. That means Sarah Jerin. I am not. Uh, the question is not clear to me actually. What do you mean about that? Okay. So then, what was I'm telling? So the today's topic is the what is the ultrafiltration in the nephron. So that's why there are four sample. Sample 1, sample 2, sample 3, and sample 4. Okay, to show the result, actually what happens in the nephron. Then there is also flow rate, and this flow rate is to measure how much water is in the tubules. Okay, so now how will we understand? Like if the flow rate is if the flow rate falls from 100% to 50%, so on that time we have to understand that 50% of the water has been passed back into the blood or reabsorbed. Okay, and then to make it more easier, the dissolved concentrations, the dissolved solutes in the glomerular filtrate are represented by some of the letters like A, B, C, D. And you can tell relative concentration at different points like 3C, like C and here 3C. It means if it is urea, it means the urea is now 3 times more concentrated than it is C. Okay, so just by this way, we will try to make understand these things. Okay, now, so sample 1, sample 2, sample 3, and sample 4. Let's talk about sample 1. Look at sample 1. In the blood plasma, the blood plasma contains different types of solutes, different types of substances, including protein, glucose, urea, sodium ion, etc. So if we so protein is represented here by A, glucose B, urea C, and sodium ion D. So must see and must watch. This sample is in glomerulus. See, I just mark here. This is in glomerulus, the sample 1. Then look at sample 2. So already I have told you what is the part of the nephron. This is Bowman's capsule. So in Bowman's capsule, the flow rate is 100%. Flow rate is 100% and the protein is nil. Why? You may know the protein molecules are very large to pass through the tubules. That's why in the Bowman's capsule, the proteins is nil. And the glucose, urea, and sodium ion are in the same concentration. Okay, now look at sample 3. Sample 3, here the flow rate is 20% from 100%. That means you understand 80% of the water is reabsorbed in the blood. Okay? And then protein nil and glucose also nil here. And the urea is 3C, 3 times more concentrated, and sodium ion the same, only D. So what is the differences between sample 2 and sample 3? The flow rate becomes 20%, then 100%, and besides the protein, glucose also, glucose nil. I mean, before, that is after first coil tubule. I mean, in first coil tubule, the glucose is taken back in the blood. Is that clear? Okay. Now, finally, look at sample 4. Where is this sample? It is actually at the duct opening or at the pyramid. Okay, now just see, flow rate is 1%. That means 99% water has been reabsorbed already. And protein also needs 0. The percentage of glucose 0. Urea now at the last time, finally the urea is 60 times more concentrated and sodium ion double is here. Okay, so just see, again I'm just going to the summary. This topic is about what happens in the nephron especially. I mean a nephron and its blood supply. See, in sample 1 in the blood plasma, the dissolved solutes are protein, glucose, urea, and sodium ion. Okay, this is in glomerulus. And then, as we know, the protein molecules are very large. So, molecules are very large. That's why it doesn't pass out. It doesn't pass through the tubules. That's why in sample 2, in Bowman's capsule, the protein meal, glucose, urea, and sodium ion are in the same concentration. And now look at sample 3. In sample 3, the flow rate is 20%, falls down from 100%. That means 80% water is reabsorbed. Okay. And then this is first coil tubule, second coil tubule. After, then at last, yes, but one thing is that glucose in sample 3, the glucose is nil. Glucose concentration is 0 because in the first coil tubule, the glucose is taken back in the blood. And then, and finally, sample 4, the flow rate is only 1%, that means 99% water is reabsorbed, and protein, glucose also 0, nil, and urea 60 times more concentrated, and that is sodium ion double here, 2 times concentrated. So actually these about uh, the nephron in a single nephron. Okay, and you know the structure of kidney. This is renal artery, renal vein, and darker outer region is called cortex. And yeah, can you see or can you find out the what is this actually? This part here, that nephron I have drawn here already. This is actually here. See the Bowman's capsule, the first coil tubule, and long loop of Henry, and second coil tubule. So can you guess that the Bowman's capsule, glomerulus, and the first coil tubule, then the loop of Henry run. I mean the nephron runs down to the middle layer of the kidney, that is called medulla, and at last the medulla joins up to the pyramids, and finally the urine just empty here in the pelvis, and pelvis just join to the ureter. Okay, that is ureter, and at last ureter, then there is a muscular bag called bladder, and the urine is stored there, and from the bladder to the ureter, the urine passes out. You know the urinary system very well. Okay, so this about the ultrafiltration in the nephron. Okay, and we must have to remember the different parts of the nephron, or you have to know this is the filtering unit or structural and functional unit of the kidney. Okay, and so is that clear to everyone? So if you have any questions, so you may ask me through the comments, so no problem. Oh, Sarah Zerin Tasnim, my student. Oh, very good. Okay. So now see. So actually, I have through these 4 samples. by writing these, we can discuss or we can explain the filtration in the nephron very well. So thank you very much, and no more today. All the best, and in my next uh, live or video, I will be coming with another topic. Okay, so till then, Allah Hafiz, all the best.